Okay, we are live, so we're going to wait a little bit for everyone to get in. I just want to point out to all the people that I'm going to paste in the main chat for the session the link for the website describing all the details for this workshop. You will find there the links to papers and to details about Docker and VR packages, and even the link to Orchestra, where you can connect and start your own live instance or the workshop material, which you can use to follow along as Mahmoud is presenting. All right, I guess we can start. So welcome again, everyone, and welcome to this short workshop. Here we have the workshop author, Mahmoud Ahmed, from the Gyeongsang National University in South Korea. He will be showing us how to use both R and in particular bioconductor packages to integrate ChIP-seq and RNA-seq data, and that will help us to understand the function of certain transcription factors. So again, you will find the link with all the technical details in the main chat. And let's thank again Mahmoud for being here. The floor is yours.
Okay, so hello everyone. My name is Mahmoud Ahmed, and I am a PhD student at Gyeongsang National University in South Korea. So today I want to talk about an R package that I developed and submitted to Bioconductor that I use to integrate DNA binding data like ChIP-seq and gene expression data like RNA-seq or microarrays. So the basic idea here is that to find the formal way to integrate identify binding sites and integrate it with RNA-seq data to understand how the binding between a transcription factor and a certain gene can produce the effect, like an effect on gene expression. So in this workshop, I will be presenting a brief introduction about the package, and I will be going through a use case for two related transcription factors in HeLa cell to see whether they can, the two factors cooperate, like functionally cooperate, meaning they produce the same effect when they bind to the same target, or compete, meaning they produce different effects, like one factor would upregulate the gene and the other factor would downregulate the gene, for example. So the prerequisites for this workshop are just basic knowledge of R and Bioconductor, and specifically the genomic ranges package or the G-ranges object. And those three papers are related to the work that I'm talking about. The first two papers are where the model that I'm using was presented and applied for the first time. And this paper we published earlier this year that describes the work in details. So if you would like, you can participate in the workshop by following through the code in an R Markdown document. And in addition, I'll be giving a brief introduction at the beginning. And I'm really excited to have a discussion with you later at the end. So to do that, you can click on this link and search for the name of the workshop. And you will start an RStudio server instance on the cloud that you can follow through the code with me. Alternatively, you can do this locally by pulling down this Docker image and running the instance on your local machine. So the packages, the R and Bioconductor packages that I'm using in this workshop are mainly the target package and some other packages that are very powerful tools to read in data and manipulate data to the certain format that I would like to use for the specific analysis. Excuse me. So the genomic ranges package, the R-track layer to read the bit files, and the annotation DBI package to interact with the annotation packages, the TXDB, the Homo sapien HG19 genome, and then the Homo sapien organism package from the Bioconductor. And then some other tidyverse packages to read in data and manipulate it. So the general outline would be an introduction at the beginning, and then we'll walk through the code for the use case. And then we'll have a discussion and answer people's questions if they have any. So the goals of the workshop, again, is to allow walk through with the participants to read in differential expression data in R, and then read in the peak binding data from the bit files in R in the granges object, and then to run the analysis and explore the output. I hope that by providing this example, that participants can develop an understanding to the issues of integrating those two types of data, and ideally they would be able to adapt this code to their own workflow. So more specifically, I will work first with the differential expression data and the binding data to read them into our objects, and then use the biconductor packages to extract genomic information to map between different IDs, and to extract genomic coordinates for the peaks and the target genes. Then we will learn how to prepare the expression data and the binding data for the target analysis, and then I'll try to explain the tools that I added in the package to visualize and test the tools. So this is, again, more specifically, we will use data frames to read in the differential expression data, and the granges object to read in the binding data. And then we'll extract the information from two biconductor packages, the TXDB and the organism package. And then we apply the target analysis using two functions. The first one is called associated peaks, and the second one is called direct targets. And then there are two other functions in the package that I added to explore to be able to visualize the data and formally test them. So the next step would be to go through the introduction. First, a brief background about the problem that I'm trying to solve with this package. So we know that transcription factors bind to regulatory regions, such as the gene promoters, and the effect of this binding would be the induction or the repression of the gene expression. We also know that the transcription factors bind to share their binding site with other factors and cofactors and DNA binding proteins. And then by integrating, by finding the overlapping binding sites between two or more factors, and then quantify the effect on the gene expression in terms of gene expression, in terms of differential expression from a perturbation experiment, such as knockdown or overexpression, we can infer a combined function in terms of cooperation or competition. So to do that, I used the model that was proposed before by Tang et al. In 2011 to model the binding sites of to model a regulatory potential for transcription factors by discounting the distances of the peaks that are identified from the ChIP-seq to the transcription start site. So we call this the peak score, and it's the distance from the transcription start site to relative to a certain distance. And this would be the formula that we use to calculate that. So the reason this paper chose this particular formula is that this calculation would approximate the shape of the empirical observations that they made using different types of data. So visually, it would look something like that. If we put distances on the x-axis and put the score that we calculate in the y-axis, we will see that if the transcription factor bind very close to a transcription start site, it would have a higher score. And then as we go away from the transcription start site, we will start discounting these peaks. And to model the regulatory potential of genes, we collect all the peaks that are near the transcription start site for a certain gene, calculate the score for them, and then sum these scores to give a unified score for individual genes. And then here we will see that the regulatory potential of a specific gene would increase with the number of the binding sites, as well as the distances from the transcription start site. So visually here, again, if we just choose if we assign random distances and random numbers of peaks, we see that the genes that have higher number of transcription binding sites will usually have higher score. And to integrate those two types of data, the binding and the expression data, we rank the genes by regulatory potential and by the differential expression gene statistics, such as a fold change from a perturbation experiment, and multiply these ranks to get a ranked product. And this ranked product will tell us how important the gene is in this particular experiment. So visually, this would also look something like that. We have the gene scores, the gene scores from the discounted distances on the x-axis, and we have the t-statistics, or the fold change from the differential expression, on the y-axis. And then we see here that for the genes or the targets that have a very low score and very low differential expression value would be less important, and the ones that have higher distances, higher scores, sorry, and higher differential expression value would be more important. I extended this a little bit further to work for two factors instead of one. So if we have two transcription factors, X and Y, and we perturb them in separate experiments, we get differential expression compared to the controls without the perturbation. And then multiplying those two statistics, such as the fold change, will give us a regulatory interaction. And then we can use this regulatory interaction to actually find out what if the two genes, the two transcription factors share a binding site, whether they would cooperate, meaning they will produce similar effects, or they would compete, meaning they would produce opposing effects. So this was to rank the targets from the most important to the least important. But to aggregate the effect of the binding events, I'm using the empirical cumulative distribution function to for groups of genes that are, for example, upregulated or downregulated, find the proportion of genes in each category that are ranked at or better value, better x-axis value. In this case, the regulatory potential. And then we can compare this curve to either a theoretical distribution or to another curve, such as comparing the upregulated to the downregulated groups. So the four functions in this package are the associated peaks to find the peaks within a certain region and calculate the peak score. The direct targets integrates the peak score, the gene scores, and then with the statistics from the differential expression. And then plot predictions and test predictions to visualize and test the predictions that we make. So there are different approaches to integrate gene expression and DNA binding data in R. Some examples of the existing packages are listed here. So the limitation of this analysis is that we need specific kind of data for two factors in the same line, in the same cell line, to be able to make specific predictions. And this analysis assumes that the interaction between those two proteins or two transcription factors is linear, which is not always the case. And that there are some forms of binding that we can't detect, such as assisted binding. So this package is available in the Biconductor already, and there is a Shiny application that can be used locally or can be used on the web as well. So I'll now go to the code walkthrough. So if people are ready, maybe I will give people like a few seconds to start their instance if they haven't already. Okay, so in this use case, I'm using data for two transcription factors that are called yin-yang1 and yin-yang2 in a specific cell line called HeLa cell. So I will start first by loading these packages. And while they're loading, I will tell you a little bit about those two transcription factors. So the two transcription factors belong to the same family. The first one that was discovered is called yin-yang1. And this factor is a zinc finger protein, and it produces its effect on gene expression by deacetylation or acetyltransferases of stones around the promoter area. And it is known from experiments that it induces and represses several genes. The other related protein here is called yin-yang2, and it's also a transcription factor, and it's also a zinc finger protein and arose by retrotransposition of the yin-yang1 gene on chromosome 14. So in this analysis, what I'm trying to do is to answer the questions: do these transcription factors share the same targets, or at least a significant number of targets? And if they do, what are the consequences of the binding of these factors, either on their specific targets or together on the shared or the common targets? And then whether we can infer those two factors cooperate or compete to produce a certain effect. So the data sets I'm using here are publicly available data sets, two Chipseq data from the HeLa cell line for the individual factors, and then gene expression data of the knockdown of each factor separately in the same cell line. So the data processing was done outside this workflow by those two databases. The NocTF is a database to collect publicly available gene expression data where individual factors were knocked down, and then compare that to the control. And then the Chipseq data is from the ChIP atlas. And then I'm using the human genome 19 to extract the genomic coordinates and to map between different identifiers. Okay, so to download the data, you can just click here, and the data will be downloaded in the vignettes file. If you click here on the data, there are 4 files that we'll be using in this workshop. So the steps of the data analysis are first preparing the 3 sources of data, the gene expression, the DNA binding, and then the genomic data, and then to predict the specific targets of individual factors, and then to predict the combined function of the two factors on the shared targets. So the first part here is preparing the binding data. So the ChIP peaks here are downloaded in the bed format, and we'll first locate this file in the data directory, and then use import bed from the rtrack layer to read the data in R, specifically in a granges object. And this code chunk here would do that. So I'll go on and execute it. And then the resulting object is called peaks, and it's a list of 2 granges objects. So we can look here to see we have over 3,000 peaks for the first transcription factor, and we have about 3,300 for the second transcription factor. So for those of you who are not familiar with the granges object, it consists of 2 parts. The first part contains information about where in the genome this particular range exists, or this particular peak exists. And then, so this is encoded in 3 or 4 points: the chromosome name, the start site, and then the end site, and we have the strand. In this case, the strand information is not recorded. And then the metadata is just a biconductor data frame that could be any number of columns that contain any amount of information about these peaks. So the kind of things that we can do with the granges object is, for example, we can use the seq names to ask how many peaks on each chromosome. And then by executing that, you see, for example, here that chromosome 1 has over 3,000 peaks. We can do more calculations on the granges object by saying, for example, calculate the width of each peak, and then I'm using summary to give us a brief summary of these peaks. So we see here that the median length, the median width of the peaks is about 200 base pairs, and this is the distribution of them. And then we can also look at the strands. In this case, the strand information is not recorded for the peaks. So to prepare the gene expression, I'm locating the file in the data. There are just tabular files or text files. I'm using read TSV to read the data in. I'll select the relevant columns, and then I'll name this object express. So this is doing that. Those are the 3 relevant columns. I'm naming them TF for transcription factor, gene for the gene samples, fold change and p-value, and then I'm selecting only the data for the transcription factor that I'm interested in. I'm going to execute that, and then this is how it looks like. So we see that they have exactly the same number of genes, and if I'm looking at the names and the order, so they are in the same order, and then we can look here quickly at how many genes have positive fold change and are at the p-value from the differential expression calculation are below the 0.01. And then we can do the same for the second gene. You can see here the numbers that are many more genes are regulated by YY1 than YY2. I'll skip this for a second and to explain those few graphs. So in this graph, I'm trying to look at, instead of looking at just the numbers of differentially expressed genes, to visualize them a little bit. So I'm using a volcano plot. So put the fold change on the x-axis and put the significance value on the y-axis. And then I'm doing that for the first transcription factor, and then for the second. Then I'm correlating the fold. I'm using scatter plot to see the correlation between the two, the fold change from both factors. And then if I execute that, you see the figure here. Many genes are regulated at a high differential expression, and then the differential expression between the two factors is very well correlated. In fact, if we calculate the Pearson correlation coefficient here, we see that it's above 0.5. So from this, I see that the knockdown of either factor in helices seem to change the gene expression of the expression of many genes, and in either direction, the negative and the positive. And moreover, these changes are correlated. And then this suggests that these genes either bind to many targets, or just simply that they respond similar. The genes respond similarly to the perturbation of either factor. So next, we're preparing the genome annotation. So the issue here is that the express object has data. The gene names are encoded in gene symbol. So we will map the symbols to the entry IDs using the organism package, and then we extract genomic coordinates in terms of the start and the end sites of each of the transcripts from the HG19T TXDB package. And then I will resize these transcripts to 100 KB upstream from the transcription start sites. So annotation DBI has the function select. You pass to it the organism package, and then you say I want data for these particular symbols. So you pass the keys themselves, and you specify that this is simple, and then you ask for the entry IDs. And then if I execute that, I'm removing the missing values, and then we say use the promoter's function to say I want 100 KB upstream from all genes, but filter those genes by the entry IDs that we have, and then add the transcript ID, the transcript name, and the gene ID. Here I'm matching the data from the genome object to the entry IDs that we have, then add this data to the genome object itself. And then if you look here, see that we have here is the mapping that we did from the organism package, and then after we run this code, this is the genome object that we have. So the gene ID and the gene sample, and we also have 100 KB around the transcription start site. So now the two objects can be merged, and we'll merge them in a new object called regions. And this is also a similar. This is also a GRanges object that contains, in addition to the genome information, the expression information. So the way I do that is run map on the express, because it's a list of two objects, and then make a copy of the genome, match the gene names from the genome GRanges to the data frame that has the expression data, and then add this data frame to the metadata of the GRanges object. And then if I execute that, okay, so we get exactly the same object with the added columns. So here are the names from the original object, from the new object, and I see here that the distance we get the correct size, resized ranges that we chose already. So now to predict the gene targets of individual factors, I'm using the associated peaks function and then the direct target functions. So the input to the two functions is very similar. We give it the peaks, the regions, and then we specify which of the metadata columns we're using as column, the region's name. And then for direct targets, we need to pass it the column name of the metadata column that has the fold change data. Then the results would be another gRanges object, and this gRanges object for the associated peaks would have the assigned peaks. So it will find all the peaks within the ranges that we selected, and then assign these peaks to the nearest gene. And then there are several columns that are added containing the scores and the ranks. So I'll go on and execute that. And then this is the object that we get. So it's again the same object within including filtering out the genes, the peaks that are outside the range that we chose. And then in addition, we have the assigned region, the distances, and the peak score. So again, the names of these columns here, and this is after the addition, after the running associated peaks. Those are the three columns the three new columns that are added. And these columns contain the entry IDs, the distances, and the peak scores. Similarly, we'll run the direct targets. So we pass it peaks, pass it regions, and I'm using map here because we are running it for the two factors at the same time. Okay, so this is the object that we get. Again, it's the same gRanges object with a few added columns. So these are the names of the original object, and these are the names for the new column. So we added the score rank, stat, and stat rank, and the rank product. So to determine the dominant function of the dominant function of the factor, I said we could use the empirical cumulative distribution function. But first, we need to divide the genes, the targets, into groups based on the effect of the knockdown. So, for example, we could say divide it into positive and negative change, or upregulated and downregulated change. And then we show the empirical distribution function for each. Then we can visualize the effect, and then we can test them using test prediction that are provided in the package. Okay, so here I'm showing first how the distances that we found, that the distances that we got from the transcription start site correlate to the scores that we correlated. And then here to divide the genes into groups, I'm using the cut function from base R to, based on the statistics, which is the fold change, divide these all the genes or the targets into groups, 3 groups specifically, and give them these labels and distinct colors. So the plot predictions function, you pass it the score rank that was calculated from the DT object, and then you give it the groups, the 3 groups that we just made, and their colors and their labels. So I will execute that. Okay, so these are the distances of the peaks that we identified, and these are the scores. And you see here, as expected, the peaks that are very close have higher score, and the peaks that are away from the transcription start site are discounted. And these are the two curves, and these are the curves for the YY1 transcription factor, the upregulated genes as a result of the knockdown, and the non-regulated or the downregulated here. So we see here that there is a difference between the two groups of genes. And then this is the same for the second transcription factor. So you see here that the scores for individual peaks are the decreasing function that we expect, and the closer the binding site, the factor binding site from the transcription start site, the lower the higher the score. Here the ECDF of the downregulated targets is higher than the upregulated and the non-regulated targets, and therefore we conclude that the absence of the YY1 on its targets result in aggregates on the downregulation of the genes. Of course, here the labels are switched because here we're looking at the effect of the knockdown, not the overexpression. And we see that it's the opposite case for the second factor. And then we can test that using the test predictions. You pass it the ranks, and then you pass it the groups that we just made in the previous chunk, and then we say we want to compare the down to the upregulated genes. And then this applies the KS test, calculates the t statistics, and then a p-value. So we see here that this effect is significant for both factors. So to formally test these observations, I'm using the KS test, and it compares the distribution of the two groups for equality, and then if one is away, has a big distance from or significant distance from the other one, it concludes that they are drawn from different distributions, and therefore the effect is significant. Okay, so to predict the shared targets between the two factors, I'm using the okay, so to predict the shared targets of the two factors, we apply very similar analysis. But first, we need to identify the common peaks because using the subset by overlaps function from Bioconductor. And then we pass the columns to instead of passing one item to StatsCall, we pass it two for the two different factors. This way, we can actually use this fold change to calculate the regulatory potential, the regulatory interaction. So when the first object that has the overlapping peaks, we call it common peaks, and then the object with the fold change for the two factors, we call it both regions. So this is the code to subset the peaks to find the overlap between the overlapping peaks between the two factors, and then use this in a new genomic range. And then we just giving unique names to these peaks. So the express object was a list of tables, and so we use bind rows to bind those two data frames, and then pivot wider to distribute the fold change to two columns with for each factor, and then we'll end up with single data frame. Then we need to match. We need to add this new data frame to the genome information, the genome object. So we match the two. So we match the names of the genes, and then we exclude the genes that are not included in the genome object, and then we add it again like we did before. We add the both expression data frame or table to the metadata of the genome object. So we're going to execute that. Okay, so the code to find the associated peaks is exactly the same. So we pass a common peaks, both regions for the peaks and regions, and then we say calculate on we say the transcript ID is the regions that we want to use. And then the code again for the direct target is very similar. So we pass it the peaks, the common peaks between the two factors, and then the new data frame that has the fold change for the two factors, and then the regions ID is again the transcript ID. And then the only difference here is that when we say StatsCall, we want the fold change for the two factors to be passed, and those will be multiplied and used to make the final rank. Okay. So to visualize the output, we will use a very similar technique. So we divide the targets into 2 or 3 groups. The grouping that whatever grouping we're interested in. In this case, I want to see if 2 factors produce similar effects. I will call this cooperative. I will call this the group of targets where the 2 transcription factors are cooperating, and then the opposite will be the competition. So the idea here is that if the statistics agree in sign, then the product would be positive. And then this means that the 2 factors produce similar effects on those genes that they share. And then the reverse would be true if one factor is positive and one factor is negative, then we will get a negative sign, and then we'll take this as two factors competing for inducing the opposing effects on the gene expression. So I'm also using the similar graphs to visualize the output. So this is the output to see how the distances relate to the scores, and then this is to make three groups. So I'm using cut again to cut the regulatory interaction into three groups and passing them the labels that we just made. And then plot prediction, we pass it the score rank, the groups, and then the labels and the colors that we made. So we get a similar graph here. It shows us the relation between the distances and the peak scores, and then we get similar curves here for competing and cooperating targets. So we see here that even though there are many targets where the two transcription factors compete, the targets where they cooperate, they rank higher in this case. So this observation can be tested using the KS test. So what I'm doing here is say test prediction on the scores and pass it the groups, and then comparing the cooperative targets to the non-regulated targets. So if I execute that, I get here a significant distance between the test is saying that there is a significant distance between this curve and this curve, more than what you just expect by chance. And then if we compare the cooperative to the competitive targets, we also get a significant effect. So the summary of this is that using this workflow, we could integrate the transcription factor binding data and the gene expression data in the same cell line. So this is based on the algorithm an algorithm called Beta that was proposed by Tang et al. And Wang et al. 2011 and 13. And then using this, I could predict the combined function of the two factors. In this case, I conclude that YY1 and YY2 cooperate. That they strongly cooperate on a few targets, even though there are many targets where they still compete. Okay, so I think the code walkthrough is done. So if we have time, I can answer questions if there are any.
Thank you so much, Mahmoud. I see already a question. So Devika Argwal asks, how do you decide that 100 kilobases is the best distance to take from the TSS to identify the peaks from the Chipseq data?
Okay, that's a good question. There is no right answer to this one. I'm using 100 kb here because I want to be more inclusive, and also because if I use a smaller number of if I use a smaller distance, I will get much fewer targets because the YY2 has very few targets, has very few peaks to start with. But yeah, I guess it depends also on what you want to calculate exactly. So if you want to see just the transcription effect of this binding, then you would use a smaller distance. But if you want to see all the effects that the transcription factor could do, then you would use a bigger distance.
Okay, I guess a follow-up question, another naive question. As the peak score you get is dependent on the distance, do you need to correct for this distance or rather this bias?
Well, if I understand the question correctly, I think the idea here is to use the distance. So maybe if you could elaborate a bit more on this.
Yeah, Davika, if you want to raise your hand, I could invite you on the stage, so maybe we can interact more easily.
Yeah, sure, that would be great.
Yeah, here you go.
Hi, yeah, I was just wondering because you showed us a plot where you showed that the peak scores decrease as the distance increases from the transcription start site. So obviously there's a bias in the scores and the distance. So do you need to correct for this bias in a way, or that doesn't matter because you have chosen an arbitrary distance?
Okay, so we first choose an arbitrary distance to include the peaks, but then we say that the peaks that are closer to the transcription start site should have higher score because they're more likely to induce an effect.
But then you're not choosing the high score peaks only in your downstream analysis, right? You're still taking the low score peaks as well.
Yes, and they are discounted because they have lower score in the final rank.
Oh, I see. So, okay, that's fine. Thank you.
Well, actually, I have one,
Sure.
which is really, really practical. So I was trying to look into your RStudio session, how much resources your analysis was going to take, since you are taking all this information across the genome. I was wondering how much RAM, I mean, the speed is of course good, but how much RAM is going to be needed for this type of an analysis?
Well, to be honest, I haven't done like a formal like profiling for how much resources it's needed, but it's not much. And this is not because the package is great. It's because the containers from Bioconductor and the packages, the annotation packages specifically, they work really, really well with very limited resources. Like I can run this on my laptop and it works fine.
Thank again, Mahmoud, for this great walkthrough for his analysis of ChIP-seq and RNA-seq.
Thank you.